For part 2 of chapter 22, we are going to discuss microevolution versus macroevolution and observations and other kinds of evidence for evolution. Remember that the book defines evolution as descent with modification. That can actually occur over short periods of time or long periods of time, and that's the distinction between microevolution and macroevolution. For microevolution, we are looking at the change in genetic makeup of a population over time, or a change in how the population looks over time, as long as the appearance of the individuals is dependent on heritable components or genes. Microevolution can occur in a single generation or over a few different generations, but it's generally considered evolution over a short period of time that does not result in a new species, just in differences in genetic makeup of the population. Macroevolution is really just microevolution over a longer period of time. So it can require hundreds or thousands or millions of generations for new species to be formed from old species. So macroevolution can include the creation of new species through transmutation, which is a gradual change of one species into another, or splitting of one species into two. Macroevolution typically is a much larger scale type of evolution than is microevolution, occurs over very long periods of time. Darwin's theory of descent with modification argues that through series of microevolution and eventually macroevolution, all species are related to each other through common ancestry. One example of microevolution is exhibited by the video you watched or will watch about Darwin's finches, the medium ground finch that was studied by Peter and Rosemary Grant. Remember that natural selection occurs when there's variation in a population. And in this image right here, you can see two different individuals of the same species. This individual on the left has a shallower beak, and this individual on the right has a deeper beak. So there's variation within the population. And if we look on this image right here, you can see that time is on the x-axis. So what we see over time is that the beak depth of the individuals is changing over time. If we can illustrate that the beak depth is changing in this population over time, the average beak depth, and that beak depth is heritable, and that is being selected against, or is a tool that is being selected at different times, then we can demonstrate that that is natural selection or changes in the population over time. Natural selection is one way that evolution can happen. It's the main way we are discussing in this course, the way that evolution happens. So evolution by natural selection is occurring in this population, but it's not creating new species in this example. So this is an example of microevolution. And this will make more sense as to what the selective factors are once you watch the video. Macroevolution can also be seen with the example of the Galapagos finches, where we have one ancestral finch from the mainland of South America, and evidence suggests that population of an ancestral finch was blown off to the islands, and eventually over long periods of time, thousands of generations, has evolved and split into all of these different species. In fact, there are 13 different species of Galapagos finches. And again, the finch video that you'll watch with this lecture will explain a lot of that. But this would be an example of macroevolution, where we have one individual species that if given enough time, results in multiple different species that are related to this one common ancestor. Now we're going to talk about evidence for evolution, and the first category of evidence for evolution was direct observation. One great example of a direct observation of natural selection in action is if we look at organisms that adapt to new species that are introduced. So here's an example of a soapberry bug right here. These exist in Florida. They are native to Florida, so they exist there. And they originated in southern Florida, and they feed on a type of plant called the balloon vine. Soapberry bugs that live in southern Florida and feed on the balloon vine have very long beaks that they use to insert into the fruit and access the seeds. In the balloon vine, the seeds are pretty deep within the fruit, and so the long beak on the bug is necessary to get to the fruit. There's an introduced species of tree called the golden rain tree that has fruits that the soapberry bug can also access. The golden rain tree is found in central Florida, and soapberry bugs that feed on the golden rain tree were predicted to have slightly different types of beaks, feeding mechanisms, for eating the seeds of the introduced golden rain tree seeds. In the golden rain tree fruit, the seeds are much closer to the surface of the fruit, and so scientists predicted that the beak of the soapberry bug found in central Florida accessing the golden rain tree fruits would have a shorter beak. So let's look at the results of this study. On the x-axis, we have beak length. So this is how long the beaks are. That's for both of these figures A and B. On the y-axis, we have the number of individuals. So this will tell us how many individuals have these different length beaks. And then the difference between A and B is in A, we're talking about the soapberry bugs that are found in southern Florida and feeding on the native balloon vine. And the second one right here, B, is looking at the soapberry bugs that are found in central Florida on the introduced species, the golden rain tree. And these marks right here are indicating the average beak lengths of soapberry bugs found in museums collected before the introduced species in central Florida. Before the golden rain tree was introduced in central Florida. So before it was introduced, soapberry bugs found in both southern Florida and in central Florida had beak lengths that were very similar to each other, around 8 3/4 millimeters. In southern Florida, where the native species still exists and soapberry bugs still feed on the balloon vine native species, the mean beak size is pretty similar to what we see in the museum specimens. Maybe it's a little bit longer than museum specimens, but pretty close. When we look at the beak length in central Florida after the golden rain tree was introduced and the soapberry bugs have been feeding on those for generations, we see that the beak length of these individuals is much smaller, which was predicted by the scientists doing this research, because the seeds are much closer to the surface of the fruit in the golden rain tree fruit. So remember how natural selection works. We can anticipate that a normal population would have looked like this in terms of beak length when the introduced species of golden rain tree arrived in central Florida. But individuals who had larger beaks would not be very efficient at feeding on the fruit. So many of the individuals with the larger beak sizes probably did not do well and maybe didn't survive to reproduce. The ones who did survive to reproduce were the ones with the smaller beaks, and therefore they were able to survive and reproduce better, and the resulting generations had smaller beaks because of natural selection. Again, this is assuming that there's variation, which we can see there is variation in beak length along this x-axis. We assume that beak length is genetic. There's no information here specifically saying that, but we assume that beak length is genetic, and the selective pressure in this case was the depth of the seeds within the different fruits, and it was acting on the beak length of the soapberry bug. So this is an example of direct observation of a change in populations based on an introduced species in Central Florida. This is over a very short period of time, over the last couple decades. Another great example of a direct observation of evolution is an antibiotic resistance. Antibiotics kill or inhibit the growth of bacteria. Antibiotics are actually produced as defensive mechanisms by microbes. So, for example, penicillin was first discovered on moldy bread. That moldy bread actually inhibited bacterial growth, and the antibiotic aspect of penicillin was produced by the mold itself. Bacteria can accumulate mutations over time. They have very rapid reproduction, and mutations can accumulate through a lot of reproduction, and also bacteria can share their genes with each other through plasmids. So mutations and horizontal gene transfer result in a lot of genetic variation within bacteria. Sometimes a mutation may allow a bacteria to break down a drug or to counteract a drug's effect in some way. Because we often are prescribed antibiotics for viral infections, we overuse antibiotics, the drugs that are meant to kill bacteria. Another thing that we often do that is really bad in terms of bacteria requiring resistance to antibiotics is that often when we are prescribed antibiotics for a bacterial infection, we don't take all of them. We stop taking them as soon as we start feeling better. And that can contribute to the problem of antibiotic resistance in bacteria. For example, we might take several doses of antibiotics, and then what that does is it kills off the bacteria that are easy to kill, but the ones who would have taken a little bit longer to kill, they're still alive, and when we stop taking the antibiotics, those ones that have a little bit of resistance start multiplying and again accumulating additional